Well, so I'm very delighted to present our work on a text mining system called eMIND that enable automatic collection of protein variation impacts in Alzheimer's disease from literature. This work is conducted by our team of investigators and our students at University of Delaware and Georgetown University as part of a collaborative project with the UniProt Consortium, supported in part by an administrative supplement supported by the National Institutes on Aging. At the end, I'm also going to touch upon our planned work to further enhance eMIND using large language models and generate variant impact summaries using a RAG workflow, which is a project of a newly funded NIA supplement project. So Alzheimer's disease and related dementias, AD and RD, as we know, are really important disease. And UniProt knowledge base is a key resource that deals with protein sequences and function. And through this NIA-funded initiative, we also provide curation specifically for information relating to ADRD, including curation of key target proteins, development of disease portal, and application of text mining methods to increase literature coverage. And so for this talk, I'm going to focus on the literature coverage side. But in order to engage the user community, we conducted a workshop with both users and resources to identify curation target and also collect information regarding the kind of portal that we should be providing for information dissemination. So eMIND, it was developed to capture impact of variants from the literature. But what is the impact? So we define impact of variants in this case at two levels, at the disease level and at the protein molecular level. In the context of UniProt expert annotation of natural variant, again, priority is given to those that are linked to disease and impact on some property of the molecular level. And the example sentence shown here is the type of entities, the type of relations that we extract, where here we show a protein coding variant of gene presilidin 1 and its impact, in this case, the effect on the protein interaction. So the impact type that we capture here by eMIND, again, are those that are relevant to UniProt variant effect annotations. So at the molecular level, we capture different kind of impact on protein activity, interactions, post-translation modification, structural, biological processes, processing protein abundance and protein aggregation and so forth. And eMIND is also capable of capturing disease level information such as risk to disease, phenotype, and disease onset, although that's not the focus of this work. So we have to use dictionary, regular expression, ontologies for us to be able to make the data much more interoperable with other type of tools. And so, for example, the impact phrase with a protein ending interaction binding would indicate a specific impact type, and that is impact on protein interaction. And we normalize this impact type using the variable ontology. So this is the overall eMIND workflow. It's a very busy slide showing many components, but I'm going to only cover a key components here. And you are welcome to refer to our preprint in bioRxiv for further detail. So the workflow here involves, first of all, recognition of different type of impacted entities, and then detection of phrases describing the genetic variants, and then a relation extraction classifier that would extract the impact relations between the variation and the impacted entities within a sentence. So eMIND use various tools for the different components. We use external name recognition, entity recognition tools, including PubTator for concept recognition, such as gene, variant, disease species. And we also have our own tools, such as DIMEX, for connecting the mutation to disease, along with several other text post-processing tools already developed by our team. And so the relation extraction classifier is built here using transformer pre-trained BioBERT model. In the pre-trained stage here, the BERT learns the general language representation for the words in the text sequence, and essentially with 2 masked entity. And the BERT model was fine-tuned then on corpus containing impact relation of different types. In this case, impact of microRNA on disease. And again, the training set for the microRNA on disease, it was derived from Myriad, our text mining tool that detects microRNA in association with disease. And the premise here is that the structure and the words used in the statements that describe the impact of one entity on another or on events or process are not specific to the 2 objects under consideration. So our BERT model was fine-tuned on microRNA impact training examples with about 10,000 positive, 12,000 negative examples. And we conducted an evaluation on the efficacy of eMIND to extract variant impact relations with a small set consisting of annotations of impact relations from 60 abstracts covering 25 gene targets. The evaluation corpus has a total of 163 positive instances involving 69 unique variants on the different impact types. And one can see that in the results here, protein abundance, biological processes, protein aggregation interaction are of special importance in the context of ADRDs, as these are really predominantly mentioned in those disorders. The system achieved at the end a recall of 0.84 precision or 0.94 and give us confidence that the training approach is pretty good. So based on this early evaluation, some improvements were already made to the system and the annotation guidelines. So we are currently annotating larger corpus to evaluate the system further. For this, we use the TIMTAT framework developed by Zion's team with abstract pre-annotated by Pat Pater for gene mutation disease. And we have the trained annotators who are participating in this activity. To make it easier, we divide the task into 2 steps: identify the relevant impact sentences, those containing the variant impact, and then annotate the entities and relations in the sentences. And some example impact sentences are shown here. The annotations have good agreement of more than 80% when selecting sentences containing variant and impact. So for the annotation of entities and relations in sentences, the curators then have to check the annotations by Pater and then add impact types and impact type entities and create the relations between variant and gene proteins, variant disease, and variant impact, as shown in the TIMTAT interface here. No, you can't see quite well, but basically that's the interface that allows us to really mark the sentences. And so we have annotated a total of 200 abstract with this kind of impact sentence level, and also a number of abstract entity at the relation level. So the output of the eMIND is stored in a MongoDB and can be viewed in the eMIND website. Again, we have to use the control vocabulary ontologies to foster interoperability. So this includes HGVS for variants, disease ontology for disease, the variables for impact types. And we then use this type of information and the structural format to add the information and annotation to the UniProt knowledge base. The set that resulted from this study is currently already being used to train the model to identify new positive abstracts. So this process could be continue updated with new data. So the development of eMIND was really motivated by the general need of scaling up curation. And so the contribution of publications with this kind of structure and notation to the UniProt bibliography section would be greatly expanded through this type of structural information. And it can also help curators to find publications for further curation. And so the output of eMIND are presented in the tab deleted format in the UniProt bibliography pipeline. And so here you can see examples of a UniProt record. The field include the UniProt session, the source of information like PubMed ID, source of information from eMIND, the PubMed ID, and the annotation category of disease and variants, and again, with those control vocabularies. And you can go to the specific record and you can see specific examples of eMIND extracted information that has already been incorporated into the, in this case, a SwissProt entry with evidence attribution. And so there are a number of ongoing developments for eMIND. So we continue to run evaluation of eMIND with new corpus that we are curating. We will be providing regular updates to UniProt computationally mapped bibliography using eMIND. And to do this, we will be training the lead suggest variant model using eMIND abstract currently integrated already into UniProt to identify as training set to identify new abstract for the UniProt pipeline. Meanwhile, eMIND is generalizable. Again, we have developed a suite of text mining tools tailored for different purposes. And in this case, we will continue to expand eMIND to detect impact of variants on other diseases. In the remaining few minutes, I'm going to present our planned work to further enhance our text mining system using large language models to generate variant impact summaries using a RAG workflow. Thanks again to this new administrative supplement from NIA. So in this new project, we'll be developing a pipeline to automatically process PubMed abstract at full scale using eMIND. Sentences abstract identified by the BERT-based eMIND tools as containing information about the functional impact of AD-related variants will be used as input to GPT large language models to create natural language summaries. In parallel, we are going to be using the LLM to verify the accuracy of the relation extracted by eMIND and extract functional impact information from eMIND associated positive sentences and AD-related abstract in a structured format similar to those already used by eMIND. Then output from the large language model will be manually evaluated by expert curators, and we will use prompt engineering strategy to improve performance based on that evaluation. And finally, we're going to disseminate the summary and relation to the community, again, through the UniProt website and API. So with the performance of eMIND already reaching precision of 0.94, we're going to use large language model as independent method to verify the eMIND relations. So we're going to provide LLM with the variant, the evidence sentence we have, and the type of impact detected according to eMIND, and ask whether the variant is associated with the given impact type in the given sentence. We will use scripts and the LLM API to fully automate this validation process and manually evaluate whether the combined eMIND LLM system gives better performance than the eMIND alone. Then next, we're going to see whether LLM can extract variant impact relations on its own. So in this first implementation, we provide eMIND positive sentences as the context. And in the preliminary study shown here, we conducted prompt, including through three eMIND positive sentences that describe impact of this particular variant in APP, and ask LLM to output a table showing the variant and its impact. The results are really promising. The table is complete and correct. And so we're going to further engineer prompt to try to include impact type, express as variable term, and the PMID in the output table. So if the performance at sentence level is good, then we can further expand to more extensive text as the context. And the GTP LLMs, again, capable really to summarize information from multiple pieces of text into a highly readable summary. And so these are the projects here that we are developing. Is we're going to again provide free text summary using of the functional impact of AD-related variants using LLM, and we are already using the GPT 3.5, and the result is shown here. So I think I'm going to not go into the details, but you can see that we can generate those type of information quite readily. And again, by combining the text mining and the large language model, we will be able to drastically increase the confidence and also the coverage of this type of information. And with that, I'm going to conclude by acknowledging our amazing text mining team from both Georgetown University and University of Delaware. Thanks to our eMIND consortium collaborators and funding from NIH. With that, thank you.